Hello and welcome. This is Mr. Buss, and I'm going to go through the 6B RNA and Proteins notes, similar to what I would have done in the classroom. Obviously, with distance learning, I made some revisions and did things differently. But so it's kind of like a worksheet that you fill out, but I'm going to try to go over the content so that you get it, not just from the videos from like the Amoeba Sisters and Bozeman Science and stuff like that. So I want you to be able to get it from me as well. So here we go. Let's go ahead and get started. RNA, ribonucleic acid, is very similar to DNA, but there are some, distinct differences, and so got to be familiar with those. First of all, RNA is single-stranded. Remember, DNA has two strands running anti-parallel, right? So they're running opposite of each other, two-stranded. And not only that, but like RNA is just like a small little segment of the DNA code that's been changed into an RNA code. So, I think of like DNA as like a huge novel or something like that. And then RNA would be just like, like a paragraph or a page from that book. Just a small little segment of it. Okay, so and one benefit of that is that RNA is small enough that it can leave the nucleus. DNA being double-stranded and a longer sequence cannot leave the nucleus. It's in eukaryotic cells, at least. Remember, you are made of eukaryotic cells, plants, animals, fungi, and then like prokaryotes, bacteria, those types of things are they do not have a nucleus. Okay, so for them it's different. They have circular DNA chromosome, and it's just inside the cell. The nucleotide itself for RNA has some differences as well, and similarities. So again, there's a phosphate group there. There's a sugar, although it's a ribose sugar. I noticed that there's an OH on the ribose sugar. Here's a DNA nucleotide on the right, and there's an H. So that's just kind of what I noticed there. Deoxyribose, there's no oxygen on it. The oxygen is left. So just very similar, but subtle differences there. And then adenine, cytosine, guanine, and uracil. So instead of thymine. So, for DNA, apples to trees, and for RNA, apples under the tree. A to U. C to G still. And now this picture, I think, is fantastic. It's got a lot going on, though, so don't be too overwhelmed by it. We're going to kind of break it apart a little bit further down in the document as well. So this is just kind of an overview of the process, where, why, and how, and then we'll get to more specifics, more AP Bio kind of specific content. The where, well, first I'll say it's kind of, even though there's two names, transcription and translation, it's really one continuous process, right? There's a protein that needs to be made, and it starts with the DNA, as you can see here, in the nucleus, kind of getting separated apart, similar to a DNA replication, and then it's not shown, but like then an RNA polymerase would hop onto one side of the DNA strand, happens to be this side, so I'm going to call that the DNA, this is the template. I'm going to call it the template because it's the side that is matching up to complementary RNA nucleotides. This side, nothing doing. It's just had to get split apart. And which side how do you know? You don't have to know that. You just know that one side is a template. It'll tell you in a problem. This is the DNA template side. This is the side that's going to be transcribed in your body. The code would determine it, right? So your body would know, and the machinery in your cells would know because of the code. Okay. So that DNA is going to open up. RNA polymerase is going to go in there, zip through, and make RNA, and then that RNA is going to leave. It's going to be called pre-mRNA because it's not modified yet. It has to go through some modifications that we'll talk about in a couple minutes, a little bit further down the page. Once it gets modified, it's going to leave through a nuclear pore. Okay, and so that's the end of transcription. So transcription is the process in the nucleus where DNA is coding for RNA. And then translation is the process that happens at a ribosome outside the nucleus. So we learned about ribosomes in Unit 2. We learned that there are free-floating ribosomes. That's all that prokaryotes have. And a lot of our ribosomes, because we are eukaryotes, are on the rough endoplasmic reticulum. And then right outside the nucleus is like surrounded by rough endoplasmic reticulum. So as soon as the mRNA is going to basically diffuse out of the nuclear pores, then it's probably going to have a ribosome attached to it. The ribosomes, I think they look like a hamburger bun, okay? There's two sides to it. There's this smaller side down here, this subunit, and then there's this larger subunit on top, okay? And they're not associated unless they are working and bound to mRNA. So like in your cell, they'd be kind of disassociated. And then if I'll use my kind of pencil here, if this were the mRNA that left the nucleus, it would, and I'm just kind of like glossing over the details here, but it would bind to one of the subunits, and then the other subunit would lock on a place, and then it's going to rip across that, and it's going to translate that sequence into an amino acid sequence. So let's just take a look at that process a little bit. 3 RNAs come together for this process. mRNA, the messenger RNA, right? That's what we talked about. That's this right here, the messenger RNA. rRNA, which is the ribosome. Ribosomes are made up of rRNA, ribosomal RNA, and proteins. So it's kind of a mixture of protein and RNA that's bound together to make the ribosome. And then the third one would be this right here, tRNA. And I call it transfer RNA because it transfers an amino acid to the growing polypeptide chain. All right, so the brief glossing over this, but just kind of go through a little bit. The ribosome has 3 rooms for rent. 1, 2, 3. Okay, and as this thing moves across the mRNA, a tRNA will come in, match up. How does it match up? Well, look at the code, right? The mRNA has these codes. Remember, they're called codons, and they're triplets. So like here's a triplet. Here's a codon. Here's an mRNA codon. Here's a codon. So like, you know, in the English language, a word can be 1 letter. It can be 10 letters. Variable. But for the genetic code, every word, I'll call it a codon, is 3 letters. Okay, so sometimes they're called triplets too, codon or triplet. And that's it. It's got to be 3 letters because that's how the tRNA works, is it reads a triplet. So a tRNA will come in, so you can see this one's coming, it's going to match A to U, C to G, and G to C. So this one's coming in. It hasn't entered yet. When this spot vacates, it'll enter. This one is already matched up. Okay, so the tRNA matches up to the mRNA, and the ribosome holds it in place, and then the amino acid gets smashed together next to another amino acid. Well, guess what? It's going to go through that dehydration synthesis reaction where they're pressed together. A water molecule will leave, and a polypeptide bond forms between the 2 amino acids that are wedged next to each other. Okay, and so that's happening. And then when it's gone through that process, the tRNA will get the boot. And guess what? This tRNA has lost its amino acid, so it's going to have to reattach and find another amino acid for it to like reactivate. Okay, it's going to find a specific one based on its specific code and structure. I will mention there's a term here called anticodon. So this is the codon. The tRNA, since it's complementary but it's not a codon, they call those the anticodon. Sorry, I'm writing sloppy. I'll try to write neater here. And so what you're going to get is, as this thing moves across, as this complex of ribosome, transfer RNA, and mRNA, as it moves across, you're going to get a growing polypeptide chain or protein chain. And remember, just brief review from Unit 1, that proteins have a primary structure, and that's the amino acid sequence, like leucine, isoleucine, valine, tryptophan. It's that sequence. And then the secondary structure is folding into alpha helixes and beta sheets. Tertiary structure is probably going to happen maybe more at the endoplasmic reticulum, where it's going through like hydrophobic, hydrophilic folding, hydrogen bonding, disulfide bridges between cysteines, and other like polar, non-polar interactions. So it's kind of getting more of a three-dimensional shape with that, until finally it's a fully formed protein. It might combine with other proteins. That's the quaternary structure. So that's translation. It's RNA to proteins. A couple more things before I stop the video, and I'll probably have to make 2 videos for this. I mentioned up here that when the mRNA is initially made, it has to go through modifications, and I didn't say what those were, so I'll say that here. So 3 modifications you have to be familiar with. One is that within the gene that's being transcribed, turned into mRNA, you might have sections of it that code for proteins called exons. And they're called exons because they are the parts that are expressed. Okay, those will stay. And the introns are parts that are not expressed. They are in between the exons, and they will have to be spliced out. And I'm not going to go through the details, but there's structures and proteins involved that will grab those sequences, fold, and clamp and splice and move things around and take care of all that. So the exons will stick together. AP-Bio wants you to be aware that even though in this picture I showed that they kind of all lined up, occasionally they might not do that. This one might end up over here purposefully. This one might go over there. This one might end up there. So you can have a mixing, and if that happens, obviously you're going to have more. You could have like one gene code for multiple proteins, because if the exons get organized in different ways, it's going to fold differently, and you're going to have different versions of a protein based on the way that the exons have lined up. Oh, and I'll just mention them before I turn the video off that you'll notice as well, further modification would be on one end of the mRNA, a 5' GTP cap is added, guanine triphosphate. It's like ATP, but it's not adenine, it's guanine. And that's going to add to some durability of the mRNA, so it doesn't get degraded too quickly by enzymes that are meant to do that, to degrade RNA, because it can't stick around forever. It will always be transcribed, translated. And then at the 3' end, you get a poly A tail, so a bunch of adenine nucleotides get added, and eventually those things will start to get cut off. So you add a bunch of those, and it's going to give the mRNA durability as well, because it'll take a while for all those A's to be degraded before the gene sequence gets degraded. So just some modifications there. All right, I'm going to pause the video because I feel like I'm getting to that 10-minute mark, and then I will start the next one. I'll go through the last part of this.